Today we're talking about what happens when we violate the assumptions of Hardy-Weinberg equilibrium. So, if the assumptions of Hardy-Weinberg equilibrium are not being met, populations will change in different ways. Sometimes this just means that the core math of Hardy-Weinberg equilibrium doesn't work the way we would like it to. Other times it is a clear indication that a population is evolving, and this is really on a case-by-case basis. You have to observe the specific population in greater depth to figure out exactly what's going on. And there's lots of reasons for this, like in a small population, excuse me, in a small population, random chance can eliminate alleles or allow them to fixate more easily. Migratory individuals will remove their alleles from a gene pool and bring them to others. Mutations add new alleles to the gene pool. Natural selection gives individuals advantages over each other, so they get to reproduce more and contribute more genes to the next population or the next generation. And non-random mating does a similar thing. It's just instead of an environmental advantage, it's just, hey, people like this one more. They're going to breed with it more. So let's take a look at each of those in greater detail. Genetic drift is a specific type of genetic change to a population that's just driven by random chance. And small populations are the ones that are most susceptible to this. If you have a population of 20 individuals, if you lose one individual, that removes 5% of the alleles from the population. It doesn't matter how common or rare alleles are in that population unless they're completely monoculture and uniform for every single allele. Losing 5% of your alleles is a huge change to any allele frequency. So small populations are really prone to this genetic drift. A very common university lab people will do is doing a fruit fly lab where we just repeat generations of breeding fruit flies, and every generation we take between 5 and 8 flies and use them to start the next generation. And what you find when you do that experiment is that you either lose certain alleles or you fixate certain alleles, which means if its allele is fixated, it means it's become the only allele in that population very, very quickly. And it's not driven by evolution at all, because if you do this again and again and again and again, you find it's completely random which one fixates and which one gets lost. So genetic drift is often caused by natural disasters, human interference, and habitat destruction. Anything that just comes in and interrupts the population, especially if it kills off a lot of individuals. But this can happen randomly too. The diagram I have here, you go from 75 and 25% of certain alleles to 71% and 29%. Why is it? Well, it could just be that that allele happened to randomly get into more sperm and egg that generation. And with a small population, that's easy to do. But with a big population, it's really, really hard because law of averages, everything evens out over time. You can fix genetic drift if you are humans and you like to muck about in other populations through reintroduction programs. And this is often what breeding programs at zoos are meant to do, at least the ones that put conservation ahead of entertainment. So what they'll do is they'll breed animals, usually not actually at the zoo, at an off-site location. Many zoos actually have farms, which is not where they're churning out animals like a puppy mill, but what they're doing is they have large spaces and conservation programs, and they're breeding the animals, and they'll actually coordinate. There's networks behind zoos that try to ensure genetic diversity within their populations, and the goal of these programs is actually to reintroduce some of that genetic diversity back into the wild to help protect populations that have been harmed by human interference. A specific type of genetic drift that we commonly discuss is called the bottleneck effect. And what the bottleneck effect is it's a type of gene drift where a large population is rapidly reduced. Oftentimes this is a result of, again, natural disaster, human interference, or habitat destruction. But it can arise in many different ways. So in a bottleneck, it's actually named for the fact that the population looks like a bottleneck. It goes from very big to very small if you graph it out on a double-sided graph. It also works as a good analogy, though. The bottleneck means that only a couple of individuals can escape the bottle when you shake it. So the metaphor is, if you escape the bottle, you get to survive and form the next population. But what ends up happening is who gets to survive is completely random. This isn't talking natural selection here. This is talking like indiscriminate. Lots of the population dies off very quickly. So the survivors are random, and the chances of them representing the population that originally existed is very, very, very low. So oftentimes you'll get some alleles that are dramatically reduced and some alleles that are all of a sudden really, really common within the rebuilding population. And that, by its very nature, changes the gene frequencies. Some are overrepresented, some are underrepresented. And this is a very common concern with organisms that are driven close to extinction but manage to jump back. So California condors are a great example of this. They were very close to dying off. At one point, I think it was 20 individuals were left. It might even have been fewer than that. They have rebounded now. And there's actually way too many cases of this where we've nearly driven something to extinction through habitat destruction, and we managed to stop it in time that they made a recovery. But that recovery isn't even if the population comes back up to its original size, they have lost genetic diversity. So there's not really a good way to help with that, and loss of biodiversity means loss of resilience to environmental changes. There have been some scientists proposing this is what genetic resurrection technology should be used for, which is actually referenced directly in Jurassic Park, bringing back species or individual, or trying to bring back genetic material of an individual to reintroduce that through a breeding program into the original population. But so far, while that's a really innovative and cool idea, it remains hypothetical. Another type of genetic drift we deal with is founder's effect. And what founder's effect is when you have a small portion of a population break off and form a new population on their own. A group of individuals migrates out of one population, it forms the new population, and just like in a bottleneck, the new population probably doesn't represent the original population really well. A very famous example of this is actually polydactyly in Amish communities, because in some Amish communities, having 6 fingers is not common. It's still pretty rare, but it's way more common than in the rest of North America. And the reason for that is that there actually wasn't a 6-fingered person in the founders of the original colonies, but there was someone who was a heterozygous carrier for that gene allele. So over time, that allele, because that particular community doesn't interact as much with other human populations, at least in terms of reproduction, that means that that allele within their gene pool is much more common than it is in everyone else, just because you had that one person who contributed a single allele when their numbers were very, very low, and now it has just spread much further than you would normally expect it to.